Okay, good morning, everyone. Today we're going to finish off on steroid hormone action and AIS, androgen insensitivity syndrome. In case you're wondering, when I put up the topics for the lecture, I view it as a continuum. I don't always get to the last few topics of that given lecture, but I always hit them in the very next lecture. So view the board here as a continuum of topics. And then we're going to hit cell surface receptors, overview of glucose homeostasis, and insulin receptor action, if we get this far, glucagon receptor action. Also, you'll notice as you go through the reader, there should be questions in there. You should look at them because it'll give you a test your own ability to understand the material. And they're in there designed specifically for that. We'll go over one potential question today. Okay. Just to review what we talked about, some of the highlights. Hormone action can be described as binding of the hormone to the receptor, a change. As a result of this interaction, there's a change. And this change is receptor interaction with cell components, which differs from receptor to receptor. And that change induces a response. And we briefly talked about the differences between endocrine, paracrine, and autocrine pathways. That is simply differences in the origin of the hormone, whereas the target cell expresses the receptor. And that our point of view is really from the target cell, more than from where the hormones come from. Now, something I alluded to, didn't quite mention, but came out of discussions in office hours. Hormones are a broad class of molecules. They can be steroids, proteins, peptides, amino acid derivatives, of which we'll mention a lot of those. Hormone-like and lipids, gases work by hormone-like mechanisms, and as does certain kinds of environmental stress, such as heat shock, osmotic shock, can induce hormone-like pathways. Even light can induce a hormone-like pathway. We'll allude to that probably when we get to it on Monday in a different context. In contrast, receptors are proteins. Some of the proteins are transcription factors, as in steroid hormone receptors. As you'll see later today, some of the receptors are enzymes, such as insulin receptors, cell surface receptors, but they are biologically active proteins. And they are encoded by genes, RNAs expressed. You can have proteins. Mutations in the receptor genes can mediate lots of different kinds of disease states, okay, as well as kick on risk for cancer, things of that sort. So they are just genes that encode a protein that we call a receptor. Okay, keep that in mind. We then talk about homeostasis. Even though you're maintaining a steady state, it's really a dynamic response to stimuli. We talked about negative versus positive feedback. An important negative feedback you'll see next week a lot is the hypothalamus anterior pituitary axis, where the hypothalamus secretes hormone 1, induces the anterior pituitary to secrete hormone 2, which hits an organ or a gland, which makes hormone 3, which then gives you the response in the body. But hormone 3, if it gets too high, you don't want it too high, feeds back and turns down the anterior pituitary's production of hormone 2 and turns down the hypothalamus production of hormone 1. When hormone 3 gets too low, you go below a threshold of some sort, and then you kick on the synthesis of hormone 3 again, or the pathway that induces hormone 3. So this way, hormone 3 comes up, too high, bring it back down, too low, bring it back up. It gets too high again, you bring it back down. And you'll see this in human reproduction. You've got to understand this negative feedback system to have any sense of understanding of human reproduction. And this is why I talk about it. I did talk about it in terms of long-term stress. Hormone 1 is corticotropin releasing factor. Hormone 2 is ACTH, adrenocorticotrophic hormone. Hormone 3 is cortisol, which is a glucocorticoid. You induce it as part of the anti-inflammatory response, then glucocorticoids or cortisol dial down the anterior pituitary and the hypothalamus. So the question I have for you is, which organs express receptors for cortisol? Just based on that diagram. Adrenal cortex, that would be the one incorrect answer, unfortunately. What's the receptor? It's the target cell. What's the target cell for cortisol in this negative feedback system? Name one target organ of cortisol. Hypothalamus, correct. What's another target? Anterior pituitary. So the reason the anterior pituitary and the hypothalamus respond to cortisol because they have receptors for it. Which tissue type has receptors for ACTH? Adrenal cortex. There we go. So when I have an arrow going to into an organ or gland, if ACTH is hormone 2 and the organ or gland is the adrenal cortex, it has the receptors for hormone 2. Which tissue has the hormone receptor for CRF? Which one? Anterior pituitary, right. See, so the whole negative feedback system is mediated by receptors expressed to respond to the hormone. This will be absolutely crucial in understanding human reproduction, very true for long-term stress response. As I said, you are your hormones. And it's because, not necessarily of the hormone, it's important, it's a signal, it's because of the receptors. And that's my philosophical viewpoint on it. Okay? Questions on negative feedback? Positive feedback, I give you an example of labor-induced contractions. You'll see another one next week in part of the menstrual cycle as you kick on right before ovulation. You get the LH surge, but we're not going to worry about that till next week. It's another example of positive feedback. There's also a situation where it's really no particular feedback. You're just responding to the environment, and that's the rapid stress response. Some sort of stress, the example I gave you is you sit down and get ready to take your exam. They're handing out the exams, your heartbeat is going fast and everything. What happens is you feel that stress, the hypothalamus through nerve interactions, interact with the spinal cord, through more nerve interactions, it interacts with the adrenal medulla, not the cortex, but the medulla, converts tyrosine into epinephrine, or known as adrenaline, and you kick on your response. Because it goes through nerve impulses, it happens very quickly. It's also part of the response if you're walking across the street and all of a sudden some car is barreling down on you and you got to like get out of there very quick. This quick response to that is due to this part of this pathway. Same kinds of things. Because it's nerve impulses, it's extremely quick type of response. Okay? Then we talked about agonists versus antagonists. You should know the difference. Agonists are the hormones themselves, induce the response. Antagonists block the response. Very medically important. And those of you getting into medicine later on will probably be giving prescriptions to your patients on hormone antagonists as well as agonists. Female contraceptives are all agonists, for example, whereas a lot of certain anti-breast cancer therapies are antagonists. Okay? And then we start on steroid hormone action where we ended. And that is steroids are these small, greasy, hydrophobic molecules. They go into the cell, picked up by an intracellular receptor. This action, the classical part of this pathway, which is the only part you need to know about, activates the receptor to bind to the promoter of a target gene, where it can either be a homodimer, we're not going to worry about that, but in either case, it can interact with pre-existing transcription factors, which are, as you know, proteins that sit on the DNA and have very specific DNA binding sites. This kicks on the RNA and the protein expression of a target gene, thereby giving you the response. This is kind of where we left off. Now, although I drew it as a receptor and a transcription factor, you don't need to know this except if you study this later on or take my hormone action class later on. And that is, this is really a huge protein complex. For some genes, this can be as many as 50 different proteins will build up on the promoter and talk to the transcriptional machinery. Okay? But I'm just drawing it as a Pac-Man in a circle. Now, looking at this diagram, the receptor bound to the steroid, bound to the transcription factor, bound to DNA, the interactions of the receptors with various components dictate specificity. So looking at this diagram, what are some of the interactions of who does this receptor interact with? Who does this receptor interact with? The hormone, the steroid, okay? So specificity is the hormone binding domain of the receptor. So the specific part of the receptor binds the steroid. What's another component that the receptor interacts with? I can say it louder? That was correct? Yeah, it interacts with the transcription factor. So transcription factors are proteins, so there's a protein binding domain. And what else does the receptor interact with? What else does it interact with? The promoter, but what's the generic term for a promoter? What is the promoter made of? Yeah, DNA, very good. DNA binding domain. And the combination of, there's other parts to it, but this is the fundamental parts. The combination of hormone binding domain, protein binding domain, and DNA binding domain, in other words, the interactions of here, here, and here, all dictate specificity. So a given target cell will respond to these steroid receptors based on that combination of interactions. And as we'll talk about, as soon as we talk about DNA binding, there's a syndrome called AIS, androgen insensitivity syndrome, that's based and helped us understand these kinds of interactions at a clinical level. And we're going to talk about that in a few minutes. Okay? Any questions on this? So let's just focus on DNA binding for a few minutes. The DNA binding, and we're going to really focus on two different, at least initially, two different steroid receptors. By the way, what are our favorite steroids again? Estrogen, testosterone or androgens, progestins, glucocorticoids, mineralocorticoids. Not only is our favorite ones, but that's all 5 of them. There's only 5 primary steroids. There's other hormones that work by a similar mechanism, thyroid hormone, vitamin D, so forth. We're not going to worry about those. So let's just talk about DNA binding. Here's the receptor coming in, binding to DNA on the promoter. The generic term, and it's sequence specific. So the generic term is hormone response element. Okay? That's just the generic term. If you see the word element, it tends to mean there's a specific DNA sequence that's involved. And it's certainly the case with these receptors. Just like lots of transcription factors all have, virtually all of them have specific DNA sequences. So the glucocorticoid receptor, so GR, this binds to a specific sequence called a GRE. And as you might, let me just write this a little bigger here. And this is the glucocorticoid regulated element. Okay? GRE. Not graduate record exam, but glucocorticoid regulated element. This has a specific sequence. You do not need to know this sequence, and I'm going to draw on the board. Feel free to memorize it, but you're not going to be examined on it. This is the half side, TGT, TCT. I bring this up so we contrast it, I'll bring this down in a second, to the estrogen receptor. Call this ER. Again, for the purposes of our discussion, this is not endoplasmic reticulum. This is estrogen receptor. So we almost have to transliterate this thing a little bit here. Anyway, this receptor binds to a regulated element. What do you think the regulated element is of the estrogen receptor called? The estrogen regulated element. That's right. So it binds to the ERE, estrogen regulated element. So the GR binds to the GRE, ER binds to the ERE. And again, in a sequence, you do not need to know. But I'm only bringing it up to point out that it's different. That's what the only thing you got to know, that it's different. It binds to a half site of TGA-CCT. So the difference in these couple base pairs dictates specificity between GRE and ERE. So the important thing to remember for the discussion now is the glucocorticoid receptor binds to a piece of DNA that's different than what the estrogen receptor binds to. But both of them require their specific DNA sequences. So you just got to know a GRE and an ERE are different. You do not need to know the sequence. Okay? Questions? All right. So how does one prove that this receptor can bind to DNA in a promoter? So if you have this target gene for the steroid, if this is the GR, the glucocorticoid receptor, what is your prediction about the DNA binding site in the promoter of this gene? What's in the promoter that it interacts with? Or what's part of the promoter? What DNA sequence is part of the promoter? A what? Well, a GRE, right? A GRE. See? So that what defines the receptor binding site for the GR is a GRE. If it's an ER, it'll have an ERE. So estrogen-responsive genes have EREs for the most part. There's complications out there they don't need to know about. Okay, does everybody see that? It's a specific DNA sequence. Just like whatever this transcription factor is, has its DNA binding site. Did you guys do DNA binding sites for transcription factors earlier? Right. These receptors are transcription factors. Therefore, they have their DNA sequences. If it's the glucocorticoid receptor, it's the GRE. Okay, does everybody see that? Any questions at this point? Because we're going to go into an experiment that's going to help prove this. Okay, are we good? All right. So how does one prove this DNA interaction? So let's just stick with GRs, glucocorticoid receptors, and GREs. The assay that's used quite often is called a gel shift assay. I'll give you the basics first before we see the before we look at the results. The basics is it distinguishes DNA from steroid receptor or any receptor bound to DNA. It distinguishes it by size. Okay, and I'll give you an analogy with an eraser and a water bottle here in a second. So let's just go over the actual event. So what you do is you take your piece of DNA and you radiolabel it. So this is a label, radiolabel usually, and make this DNA a GRE. Okay? So you can easily synthesize DNA to be exactly matching the binding site for the receptor. That's not an issue. Easy to do. You then add the glucocorticoid receptor in this case, and always have it plus, for these experiments, you have it plus steroid so that it's active. And then you run it out on an electrophoretic gel. But under conditions that are what we call native, which means it maintains srDNA binding if in fact it exists in the experiment. Okay? So you run the gel out, and it runs by size. So this is the larger molecules. This is the smaller molecules. Okay? Smaller molecules run further in the gel. You guys have had that, right? Electrophoresis? Okay? So let me just use an analogy here. In this experiment, what is radiolabeled? The GR, the glucocorticoid receptor, or the GRE? Where's the radiolabel? The GRE in the piece of DNA. Does everybody see that? So that's the only thing you can see. These erasers are GREs. You can see that. This is the GRE without the receptor. This is the GRE with the receptor bound to it. Let me ask you an intuitively obvious question. Which of these complexes are bigger? This one, obviously, right? So you start out a mixture, you run the gel, and the smaller piece runs further than the bigger piece. Does everybody see that? Now you can only see the DNA. In this experiment, you can only see the erasers, right? So you end up with a gel that'll look like this, because you can't see the water bottle. You can only see the erasers. Does everybody see that? That's the heart of this assay. That's it. It's a wonderful assay, and a lot of people use it, and it's out in the field. A lot of people use it. So if you run just the DNA, you'll see a band that'll run so far down in the gel. You see a band on a gel. But if you have the receptor plus the DNA, it runs not as far. So that's why it's called a gel shift assay. You shift the band to a larger size due to the binding of the receptor. If any protein binds to this piece of DNA, you'll shift the piece up. So you can take the GRE and look for binding to anything else you want to look at that you feel like doing it. Or you can look at mutant receptors, which we'll talk about in a little bit. Or you can look at mutant pieces of GREs, which we're not going to talk about, but clearly exist. Actually, we'll talk a bit of one example of that, potentially. Okay? Any questions on that? So let's do the experiment, and we'll include a very interesting surprise in the field. So these are going to be bands on gels, and you have gel shift assays. So let's just do the basic controls first. So first we run just the GRE. So we take the radiolabeled GRE, add the receptor, run the gel. What do you get? One band here. This happens where it runs this time. We add the glucocorticoid receptor plus the GRE, and what do you think you see? This should be obvious when we just talked about. Band shifts up, right? Glucocorticoid response element binds the glucocorticoid receptor. Everybody there? Now let's give it the estrogen receptor. What did we say about what the estrogen receptor binds to? What does the estrogen receptor bind to? ERE, right? It does not recognize the GRE. That could be any random piece of DNA as far as the estrogen receptor is concerned. So if we take the estrogen receptor and run it against the GRE, where would the band end up? Here or here? Down. Very good. See, you don't need eye clickers to answer questions. So let's do the other experiment with the ERE. It's about the same size piece of DNA. So then we add the estrogen receptor to the ERE, run it through this experiment. What would the estrogen receptor do to the migration of the ERE? Shift it up. Does everybody see that? Very good. And then we add the glucocorticoid receptor to the ERE. And what do you predict? Again, glucocorticoid receptors bind GREs. So if you gave it an ERE, what would you see? A lack of a gel shift. Exactly right. Does everybody understand that result before we complicate this in a moment? I like complicating things. It's good for a Friday. Okay? All right. So, now if you have this assay working, and people did, what would be one of your first thoughts in the field, thinking as a researcher? Let's take every possible receptor, every possible DNA binding parameter, and let's just run this gel out and figure things out, right? Because you've got a whole bunch of receptors to test. And one of the experiments that was done was to compare the binding to the GRE to the ERE to other receptors, such as a progesterone receptor, as an example. Okay? And so they got these kind of results. Let me just put this down like this. So here's the control experiment. Here's the GRE. Here's the GR of the glucocorticoid receptor plus the GRE. Okay, then there's the estrogen receptor plus the GRE, which we just said doesn't bind. Now let's check the binding of the progesterone receptor, the mineralocorticoid receptor, and the androgen receptor to the GRE. Okay, at the time, people were making bets, right? Does the progesterone receptor have its own regulated element, or does it piggyback on somebody else's regulated element? Same with androgen and mineralocorticoid receptors. And they did this experiment, and amazingly, based on DNA binding, what's our conclusion? That's the actual result. What's the conclusion? That the the androgen receptor, the mineralocorticoid receptor, the progesterone receptor, all recognize a GRE or GRE-like sequence. That says, even though glucocorticoids are part of the stress and anti-inflammatory response, androgens or testosterone being part of human reproductive responses, mineralocorticoids, sodium balance, progesterone, the pregnancy hormone, they all recognize the same DNA sequence. So given specificity, what's specificity? Hormone binding, protein binding, and DNA binding, right? One of the three is knocked out. To define a glucocorticoid receptor response relative to a testosterone response, if you look just at DNA binding, you would say there's no difference. But where is the difference between those? Hormone binding and what else? Protein binding, right? That's where the specificity is. And it turns out if you do look at all these receptors, they all have a different constellation of protein interactions. They certainly bind different steroids. But they all have a different set of transcription factor interactions. And this is very crucial in defining the response. So it is thought now, you don't really need to know what I'm going to say in the next few seconds here. It is thought now that the DNA binding just brings the receptors in the right location in the right gene, but it's the interaction with the proteins dictates specificity of response primarily. And that's part of where the field is at right now on that. But these kinds of experiments suggested that, in fact, of the steroid receptors, there's really only two kinds of regulated elements, a GRE for the 4 of the 5, and the estrogen response element for estrogen receptors. Does everybody see that? Does everybody see the complication here? It's a good thing I didn't bet my farm on that result. Fortunately, I don't have a farm, so I had nothing to bet. But a lot of people thought there was going to be specific regulated elements. Now people think there's a little more to that than just that statement. There's it pushes it in certain directions, but let's not worry about that. This is fundamentally correct. Okay? So what this is saying is specificity is hormone binding to the receptor, DNA binding, and protein binding. Now, another way to go about all this was there was a clinical perspective on this whole thing. And that was AIS, or androgen insensitivity syndrome. Okay, so what androgen insensitivity syndrome is you have genetic males, who have on the external genitalia, have a female phenotype. And if you look at the actual pathway, or part of it, you have androgens, or testosterone, or dihydrotestosterone, bind to a receptor and give you the female phenotype. So if you're not, give you the male phenotype. If you have defects in this pathway, the default pathway is to look female in terms of external genitalia, not inside, but outside. And these people survive. This is not a lethal syndrome. And these people survive, and you can thrive, in fact, in our society. And you could take the receptors and analyze where the defects are. One side point here is this is something that you guys in medical school or later on might actually see some of this. This is third-hand information, but I saw a seminar about 15 years ago where the person giving the talk was speculating that approximately 10% of female supermodels are actually genetic males. Okay, and the reason is the phenotype is lean, very little fat, nice lean muscles, and somewhat of an androgynous look. Okay, which if we think about it, is exactly what a lot of the female supermodels look like. It's such an important syndrome that two of my favorite TV shows, House and ER, did episodes on this. So as a clinical manifestation, you see a 15, 16-year-old girl who doesn't go through a menstrual cycle. What's wrong? There could be a lot of reasons for it. One of the things they'll do is they'll check for the genotype to see if it might be a genetic male. Because inside, it's all male, so therefore there's no menstrual cycle. Okay, so you might be confronted with this. Okay, just as an aside, I have 2 daughters, and I have framed chromosome spreads of all 4 kids. And the daughters are XX, and the boys are XY. Sometimes it does not pay to know too much about stuff like this. Okay, but how does this help us out? Well, when you look at the actual syndrome, it's actually a continuum of phenotypes. You go from very female-looking to very male, almost male, sterile males. Given what you know about the receptors, this is an androgen receptor response, where would you predict? So if you have like 50 of these individuals and you sequence the mutations, where would you predict the mutations to be? In the DNA binding domain, gives you a very severe phenotype. Where else would you see a mutation in the receptor gene itself? Steroid binding domain gives you a fairly severe mutation. Where else? Protein binding domain. Protein binding domain is a much more broader kind of phenotype. So individuals that have defects in the DNA and steroid binding domain tend to be much more female-looking. Those that are across the spectrum tend to be a little bit more male-looking because there's more subtle kinds of interactions. The most severe form also is, remember I said receptors are proteins. They're encoded by, that are translated from RNA, which is encoded by a gene, right? You can also have mutations in the promoter of the androgen receptor. The old name of that syndrome is called testicular feminization, but it's really a type of AIS, or androgen insensitivity syndrome. So it's multiple different mutations. But a lot of these mutations in the androgen receptor all map to steroid binding region, transcription factor binding, and DNA binding. So it holds water. All this molecular stuff held water as to where the important specificity is on these receptors. Okay, any questions on that? Does everybody see that? So here's, and it's interesting, here's a case we can have one base pair change. How many bases do you have? Like billions of bases, right? 3 or 4 billion, I don't know the exact number. But a mutation, one base, can make a genetic male look like a female. Okay, so it tells you if you've got the mutation, depending on where the mutation is, it can really change the outcome here. Okay? Any questions? And again, some of you guys going into medicine will probably, my guess is some of you might see individuals that will have this. Okay? All right. Shifting gears. These are all steroid receptors. Steroid receptors, if I said to you steroid receptor, what's the first thing you say back to me? Or one of the first things. It functions as a, what do steroid receptors function as? Transcription factors, right? That whole, if you looked at that receptor, that's a classic definition of a transcription factor, except for the hormone binding. Everything else is exactly what transcription factors do. In contrast, a lot of the cell surface receptors work by pathways that are there's a much broader spectrum of mechanisms. The hormones tend to be proteins or peptides, some cases amino acid derivatives that cannot get into the cell. So these cell surface receptors have to transduce the information from outside the cell to inside the cell. So if here's the plasma membrane, here's the receptor, the hormone binds to the extracellular domain. There's a transmembrane part of this, of the receptor. Can you predict a property of the amino acids of the transmembrane domain of a standard cell surface receptor, if you had a guess? It's hydrophobic. There's a hydrophobic character to it, right? Because it has to go through the membrane. And then you have the intracellular part of the receptor, which transduces the information, creating a change to then give you a response inside the cell. And this change is called, is a generic term, cell signaling response. And some of the membrane events, some of the change that takes place is we'll talk about, you can get receptor dimers or clusters for certain kinds of receptors, or you can get receptor protein interactions take place or get disrupted. In terms of the signaling, you can have receptor phosphorylation, enzyme activities. You can have protein-protein interactions, ion changes, secondary messengers such as cyclic AMP, and so forth. Okay? This is only a few. And the point is, unlike steroid receptors, where the biological activities are quite different and the receptors work very similar to each other, for cell surface receptors, there's a much broader range of mechanisms. Okay? Now, we're going to talk about two distinct receptors. It's all involving glucose homeostasis. Just to give you sort of a physiological part of this. So, how many of you had breakfast this morning? Oh, a lot more than last time. Very good. Breakfast is good for you. So those of you who had breakfast have a high level of glucose, high-ish level. So here's your blood glucose. It's now risen, or should have soon after you've eaten. And those high levels of glucose stimulate the beta islet cells of the pancreas, which are dispersed all over the pancreas, to secrete insulin. Insulin binds to a receptor in the liver, its receptor, and causes the liver to take in glucose from the blood, store it as glycogen, or use it up, as in hydrolysis. And as a result, you decrease the blood glucose. Okay? So it's a reaction to having eaten. And depending on when you've eaten, you're at different stages of this process. Now, how many of you have not had breakfast this morning? A fair number of you. Okay? You guys, assuming, especially those of you who may have stayed up all night and didn't necessarily have a meal at 4 in the morning. When I was in college, I always had like a fourth meal around 3 or 4 in the morning. So there's like a lot of all-night diners in the East Coast. They were fun. Those of you that have low blood glucose, this kicks on from the alpha islet cells of the pancreas. A hormone called glucagon. And glucagon binds to a receptor in the liver, a very different type of receptor than insulin, which then brings in, sends out glucose. So it causes glycogen breakdown, and causes the synthesis of gluconeogenic genes, or synthesis and production of glucose, to then increase your blood glucose. This is not starvation conditions. This is just your standard glucose homeostasis, eating and not eating. Okay? I bring this up so, again, you can view glucagon and insulin as having antagonistic functions on glucose homeostasis. I bring this up because we're going to talk some details about how insulin works through its receptor versus glucagon. Even though they're both involved in glucose homeostasis, the receptors are quite different. And if you understand how these two receptors work, you understand a lot about hormone action. Okay? Any questions? Any questions? Okay, good. So this is the overlay. So let's figure out how insulin receptor works. Insulin receptor is part of a class of enzymes, IR for insulin receptor, that's really a class of enzymes that's a single pass transmembrane receptor that has an enzyme in its structure. It has an enzyme in its structure. And this enzyme is a tyrosine kinase. So it would be completely accurate to say the insulin receptor is a membrane-associated enzyme, and this enzyme activity is activated upon hormone binding. Okay? Now there's some distinctions. This is the general class, and I'll tell you some unique differences about insulin receptor, but let's just talk about the general class. What happens is the hormone binds on the outside of the cell and causes a dimerization. So the receptors dimerize. And the reason this works is because of free lateral diffusion. Let me just put this over here. It works because of free lateral diffusion. And the way to think about it, well, here's just a diagram first. Here's the plane of the membrane, and these receptors can move laterally around the membrane. The way to think about it is ice cubes in a tray. Picture water. You have ice cubes floating on a tray. How easy it is for these ice cubes to go back and forth on the tray. Pretty easy. You just tap them, right, to go back and forth. If that tray was, say, a foot deep, how hard would it be to take the ice cube and keep it down to the bottom of the tray? Pretty much impossible, right? They always pop up. So these receptors, although they do go in and out inside the cell a little bit, they have a lot of very easy free lateral diffusion. So when the hormone binds, I'll put this board back up in a second, they find each other, but in the presence of the hormone, their binding interaction is much greater, so they stay together as dimers. Again, insulin receptor does something slightly different we'll get to in a second. What does this dimerization do? This dimerization itself activates the tyrosine kinase. So as a result of the dimers, you now have an active tyrosine kinase. And how does that work? Well, one way is an eraser analogy. Here's these two receptors coming together, right? Hormone binds. See that reaction? And that's active now. So just the physical, oh good, a lot of chalk. Sorry about that for those of you with asthma like I do. I'll pay the price for this later, trust me. Okay, the point is they come together, creates a change in the receptor that this enzyme is now active, where it wasn't active before. Everybody see that? So the dimerization plays a direct role in the activation of this receptor. So, what does every self-respecting enzyme must have? A substrate, right? If it doesn't have a substrate, what good is it? In this case, interestingly enough, the substrates are the other receptors, subunits. Y being tyrosine. Does everybody have the single amino acid, single letter code, right? Y is tyrosine. So what happens is these receptors phosphorylate each other at tyrosine residues. Oy. I hope I didn't break this. There we go. They phosphorylate each other. So what this reaction is basically tyrosine in the protein plus the active kinase, which is the receptor kinase on the receptor with ATP. You get tyrosine phosphorylated plus ADP, right? Everybody, does that strike some familiarity from some reactions you did a little earlier? You take the last phosphate group off ATP, put it on this receptor. So why do you have this phosphorylated form of the receptor? What happens is, on the inside of the cell, signaling molecules then bind to the phosphorylated receptor. This is a specific signaling molecule. It wouldn't really matter what it is. They can be enzymes. They can be all sorts of connect proteins that activate downstream pathways. And as a result, you kick on the response. So for this class of receptors, the pathway, the sequence of events is so for this class of receptors, hormone binds to the outside, the receptors dimerize, you activate the tyrosine kinase. This kinase is actually in the structure of the receptor. So the cytoplasmic domain of these receptors is a kinase. You phosphorylate the different subunits. They phosphorylate each other. Then these signaling molecules come up and they recognize the phosphorylated protein as a landing site. Binds to the receptor, and you activate these signaling molecules and you get the response. Does everybody see that? Before I give you insulin receptor, the two parts of this that are a little bit different with insulin receptor. Similar, but it's a little bit different. So it's a sequence of events. Okay? With insulin receptor is already in some sort of dimer. It's in a very loose dimer. What it is this transmembrane protein during synthesis gets cleaved. And then it comes back together with disulfide bonds. You don't need to know that. But it's already in a dimer. And when the hormone binds, what you form is a cluster of dimers. Go like this. Okay? Not that big of a deal. It's all the same. This clustering activates the tyrosine kinase on insulin receptor, which kicks on the phosphorylation of the receptor. Does everybody see that? So so this molecule, this half of the molecule here is equivalent to this. Everybody see that? Okay? That's one distinction. So although you do not need to know the difference between a dimer and a cluster. Later on, those of you who study this stuff will become very important. You can actually see in a lot of insulin-responsive cells the clusters. Some of the electron microscopy from about 15 years ago is like pretty amazing. You can see these guys all clustering. The other difference is that when it phosphorylates itself, which it does, it also phosphorylates a substrate called IRS1. It's not, again, it's tax time, and I know your parents are probably more worried about this than you guys are. But IRS is not Internal Revenue Service, but IRS is insulin receptor substrate. So insulin receptor substrate is a heavily tyrosine-containing molecule, and it is recognized by the receptor that it and the receptor both get phosphorylated on the tyrosine residues. Okay? IRS1, view it as an extended platform for the binding of cell signaling molecules. So some guys bind to the receptor, and some of them bind to IRS1. And then kick on the combination, kicks on the response. The way to view it is, if you ever go to a big class where you have a satellite lecture somewhere in another lecture hall, right? It's the same lecture, but you're in a different location. Same thing here, not same thing. By analogy, IRS1 is an extended platform of phosphorylation of the receptor, both of which combine signaling molecules, sometimes the same signaling molecules, both of which mediate the response. Together, mediates the entire response. So the pathway is quite similar for insulin receptor, but these are two very important distinctions to see, especially the IRS1, because that means you can have many, many more phosphorylation events and signaling molecules. And I think now there's up to like 5 or 6. There's IRS 2, 3, 4, 5, 6. Don't need to know any of that. IRS1, or insulin receptor substrate. So this weekend, when you guys enjoy your meals, think of it as regulating glucose, and think of it as phosphorylating insulin receptor and IRS1, and you'll be in good shape. Okay, have a great weekend, and we'll see you Monday morning.